Welcome. Today we are going to learn about the scientific method. We will also cover inductive reasoning and deductive reasoning. By the end of this lesson, you should be able to define key terms related to the topic of study, describe the scientific method, and differentiate between inductive reasoning and deductive reasoning. Let's look at key definitions. To begin with, the scientific method is a research method involving key steps that include observation and experimentation. Hypothesis refers to a proposed explanation for an event that one can test. An example of a hypothesis could be, the meeting room is dark because the lights are not functioning. This is a hypothesis because the statement tries to explain why the meeting room is dark, and it can be tested by checking whether the lights are working or not. An important aspect of a hypothesis is that it should be falsifiable. This means that the results of the experiment can show that the hypothesis is wrong. In our previous example, the lights could be working and the room could be dark simply because someone turned the lights off. This applies to scientific experiments as well. Scientists formulate hypotheses that can either be supported or not through results obtained after conducting experiments. One thing to remember is that a hypothesis can never be proved, it can only be supported. Another term is theory, which refers to a tested and confirmed explanation for observation. A theory is supported by strong scientific evidence, unlike a hypothesis, which is a proposed explanation. An example of a theory is the cell theory, which states that all living things are made up of cells, and cells come from pre-existing cells. A theory is evidence-based and has survived repeated scientific testing. Now, let's look at the process of scientific method. This process begins with observation of the natural world that leads to the development of a question. In our previous example, an individual might observe that the meeting room is dark. Then they may ask a question such as, why is the meeting room dark? This is followed by proposing a hypothesis, which could be that the meeting room is dark because the lights are not functioning, or the meeting room is dark because somebody turned off the lights, or any other reason you can come up with to explain why the meeting room is dark. However, remember that the reason should be one that can be tested and falsified. After selecting a hypothesis, scientists make predictions. A prediction is like a hypothesis, but it is in the format of if, then. For example, if the lights are fixed, then the meeting room will no longer be dark, or if the lights are turned on, then the meeting room will no longer be dark. Note that when asked to write down your hypothesis in class or in the lab, this format is used, that is, if, then format. Okay, now that we have looked at making predictions, what follows next is testing our hypothesis. This is carried out through experimentation. In scientific experiments, there has to be a control group and variables. A control group is the group in the experiment that does not undergo experimental treatment, but is kept under normal conditions, serving as the baseline for scientists to compare results. For example, if a biologist is testing whether a new fertilizer increases plant growth, then the experimental group is the plants that are given new fertilizer, and the control group is the plants given only water, no fertilizer included. A variable is any factor or condition that can change during an experiment and may affect experimental results. Examples of variables include independent variable, dependent variable, and constants. After conducting experiments, scientists analyze the result findings. They check whether their hypothesis was supported or refuted. If hypothesis was supported, then they report the results. If not supported, then they form a new hypothesis and repeat the experiment. Let's dive into inductive reasoning and deductive reasoning. Inductive reasoning is scientific reasoning that moves from specific observations to a general conclusion or theory. An example will be plant growth and light. Specific observations that can be made include noticing that plant A, B, and C grow tall in the presence of sunlight. The general conclusion will be that plants need sunlight to grow properly. It is best to remember that this conclusion is based on repeated observations, not absolute proof. In summary, inductive reasoning builds scientific knowledge from repeated observations. Consider a second example involving cell structure in plants. One might observe that onion cell, leaf cell, and root cell all have cell walls, and make a general conclusion that all plants have cell walls. Another example is that of inheritance pattern in genetics, whereby tall pea plants crossed together often produce tall offspring, Short pea plants crossed together produce short offspring, and observed results are consistent over many crosses. A general conclusion from this is that traits are passed from parents to offspring in predictable patterns. On the other hand, deductive reasoning is scientific reasoning that moves from a general rule to a more specific conclusion. Consider our first example of fish. A general rule is that all fish have fins. A specific case could be that a tilapia is a type of fish, and therefore, since tilapia is a type of fish, the deductive conclusion is that a tilapia has fins. In summary, deductive reasoning applies confirmed scientific principles to specific situations. The key is that inductive reasoning makes general conclusions, whereas deductive reasoning makes specific conclusions. Now let's look at our second example of green plants. The general rule is that all green plants contain chlorophyll. The specific case is that leaves are parts of green plants, and therefore, since leaves are parts of green plants, the deductive conclusion is that leaves contain chlorophyll. Our final example involves enzymes. The general rule is that high temperatures denature enzymes. The specific case is that human digestive enzymes are enzymes, and because of that, they lose activity at high temperatures. Therefore, the deductive conclusion is that human digestive enzymes lose activity at high temperatures. So today we defined key terms, including the scientific method, hypothesis, theory, control group, and variables. We looked at the steps in scientific method, starting with observation, followed by asking a question, then formulating a hypothesis, prediction, experimentation, analyzing results, and finally reporting results. We also looked at the main types of variables, which are independent variable, dependent variable, and constant. Lastly, we looked at inductive reasoning and deductive reasoning and provided examples. Understanding these concepts will help you in future biology classes and laboratory experiments. Next time, we'll look at the properties of life and organization of living things. Subscribe if this helped your understanding. Remember, biology only looks complicated until you understand the story.